Hi, my name is Shobhana N. I have a PhD in genetics and research experience in cell and molecular biology. I currently work in medical communications at Riata Pharmaceuticals, and I live in Dallas with my family, and I think they're also listening in here. Met Kate through this magic mentorship program. She's a 10th grader from Marymount in New York. She's fluent in 3 languages, English, Farsi, and French, and an award-winning saber fencer. She's inspired by Joan of Arc to fight for a cause. Kate was really fun to work with. We got paired due to my background in genetics and her interest in neurogenetics. I believe her dad and mom who work in neurogenetics inspired her. We've spent the last few weeks working through identifying a project in neurogenetics that can be done online through Zoom collaboration, not the easiest thing to do. Kate has been very enthusiastic, very respectful, and timely. She's shown a lot of strength in being independent and very resilient. Resilient because several ideas that we had early on did not work out the way we planned due to insufficient data. Kate was very open-minded and welcomed new ideas and ways to approach the project. I hope you find her presentation educational.
Thank you. As we said before, Shobhana studied genetics and works in medical communications, which was really helpful throughout this project, and her hobbies are reading and gardening. So my project focuses on cerebral cavernous angiomas and their genetic background, which we'll be getting into more in detail throughout this project. So first, let's review different types of inheritance patterns. The inheritance pattern that we'll be focusing on during this presentation is the autosomal dominant form of inheritance. And autosomal means not sex-linked, not on the X or Y chromosome, and that this trait that is autosomal dominant can be inherited by a man or a woman. Dominant means that it only needs to be expressed on one allele for that trait to be present. So for any given trait that is controlled by a single gene or that is monogenetic, the inheritance pattern can be influenced by either one copy of the gene or both. Let's say that allele lowercase d is dominant and it carries a disease. It only needs to be present once for the phenotype to be affected by that disease. In this case, allele capital D doesn't carry a disease and is recessive, and it would need to be present twice for the phenotype to be unaffected. However, when capital D is dominant and does not carry a disease, it only needs to be present once for the phenotype to be unaffected. As my mentor mentioned before, we were really looking to find an intersection between neurology and genetics. And the first way we did that was through looking at brain maps, which are a visual representation of brain activity. The brain map senses different types of brain waves in the brain and assigns them a color based on the brain activity. The main types of brain waves that are studied are delta, theta, beta, and alpha waves. Delta waves are from 1 Hz to 4 Hz, and they're usually associated with deep REM sleep. Theta waves are from 4 Hz to 7 Hz, and they're known for a minute amount of drowsiness. Alpha waves are associated with a state of calm and relaxation, and beta waves are associated with a state of attentiveness. As you can see on the diagram on the right, that's one brain, and that's the brain activity at these different levels. The green kind of represents the normal place the brain would be, but veering towards the reds shows hyperactivity, and towards the dark blues, there's levels of inactivity. And that highlights one of the things that brain maps are used for as a diagnostic tool. They can be used to diagnose ADHD, depression, anxiety, and migraines. And as you can see, the red towards the top that we saw in this brain map could be an indication of ADHD. My mentor and I were planning on looking at brain maps throughout different generations to form a hypothesis, However, we didn't have enough data for that, so we had to refocus. I found a research paper with MRIs for enough generations, and it focused on cerebral cavernous angiomas. A lot can be understood from that phrase, cerebral cavernous angiomas. Cerebral comes from Latin, and it's usually associated with the brain. Cavernous comes from the word is where the word cavity comes from, and it means filled with tiny holes. And angioma means vessel. So it means a vessel in the brain of tiny holes. So cerebral cavernous angiomas are a cluster of disruptive blood vessels located on either the brain or the spinal cord. As you can see in this artist's drawing, it's about the size of a mulberry, and it's also visible in black and white on the MRI on the right. So cerebral cavernous angiomas can either be sporadic with no connection to family history, or familial, which means they're inherited autosomally dominantly, which we discussed before. So the research paper I focused on started off with a young boy aged 5 who was taken to the hospital for epilepsy. The first row 2A and 2B show his brain, an MRI imaging of his brain at the age of 5. And as you can see in the left temporal lobe, the upper left, there's a lesion visible, a cerebral cavernous angioma. And the next year he came in, he was also taken for MRI imaging, and that image is available in 3A and 3B, the lower part of the graph. As you can see, the original angioma is not as visible and not even present, and a new one has formed lower on the left temporal lobe. The father of the young boy was taken in for a MRI as well. And in 4A and 4B, you can see the cerebral cavernous angioma that was found in his left temporal lobe. The next year, he was taken again, and another one was found, this time on the right temporal lobe. And the year after that, there was another one found. The grandfather of the young boy and the father of the father was also taken for an MRI, and this is an MRI from one session, and 6 lesions were found in this MRI. As we saw in the MRIs with the angiomas disappearing and appearing in different places, the genetic aspect of cerebral cavernous angiomas is highlighted. And scientists have found that they usually occur from a genetic mutation in CCM1, CCM2, or CCM3. So CCM1 codes for the protein CRIT1, and studies have found that at least 40% of those with cerebral cavernous angiomas have some sort of mutation in CRIT1. My mentor and I wanted to look at CRIT1 throughout different species to be able to further analyze its role. And based on this phylogenetic chart, which shows the genetic similarities between different species, we wanted to find a diverse range of species to study. We wanted to look at the CRIT1 area in all these species and then isolate the similarities and isolate the differences and form a hypothesis based off that. And we did so through a BLAST protein search. As you can see here, this is an example of a BLAST protein search. And what a BLAST protein search does, it takes the section that you want to study, and you can put in the different species that you want, and it'll put them one on top of the other so you can easily analyze the differences. As you can see here, this is not usually the way they're done. I actually found an ortholog, which holds all the different accession numbers, and I just had to click on them. Usually you would have to enter the accession numbers manually. And as you can see here, I am checking off a diverse range of species from cat to cattle to humans. And the names are in Latin. And when I click on them and press the button, they'll be put one on top of the other for easy analysis. I had a little trouble finding one of them, so I used a find and search to find it, and then I clicked on the alignment button, which aligned it for me. Usually the program doesn't usually work like this. You have to manually go find accession numbers for different species and then input them into the program. These are usually how it would work. You would put in the accession numbers, and the program takes a few seconds to work and align all the different species. So as I mentioned before, my mentor and I were planning to find the differences between the alignments, but when the BLAST search formulated, we found perfect alignment within all 7 species in this region, and there were no areas of misalignment. Among all these species, this region was perfectly conserved, and this told us that this must have been an important area, and that any mutation here would have been disastrous and caused a loss of function. Next, we went to analyze CRIT1's role through CRIT1's crystal structure. And that proved difficult because CRIT1 showed to be a little difficult to isolate. It was always with another protein. We found that CRIT1 provides instructions to fortify blood vessels bonds and limit the amount of leakage. And this is relevant to cerebral cavernous angiomas because they're a cluster of blood vessels. We, as I mentioned before, we went to look at CRIT1's crystal structure, and we found that it interacts a lot with another protein called RAP1. And in a few papers, RAP1 is theorized to work as a tumor suppressor. So just for a quick review, CRIT1 works to fortify blood vessels, and RAP1 works as a tumor suppressor. And when, so different proteins take up specific structures and using specific domains, and a mutation among those proteins would affect how they interact with other proteins and how they fold. I hypothesized that a mutation with CRIT1 would affect how it folds with RAP1, which is supposed to suppress tumors. And the folding would lead to a loss of function and lead to cerebral cavernous angiomas by inhibiting the structure of the blood vessels, leading them to be malformed. Some highlights from my magical experience are learning about BLAST searches and how to run them, and exploring the role of protein folding in the formation of disease. One of the challenges we found was not being able to locate enough brain maps and not being able to isolate CRIT1's protein. The lessons I learned was the importance of asking the right questions in research and asking questions that will lead you to something. And I also learned how to adapt in the face of different roadblocks and to continue pushing forth. I would like to thank my mentor, Shobhana, for her help and guidance throughout this project and for always being there for any questions I had. I would also like to thank everyone at Magic for this incredible program, as well as my ISR teacher, Ms. Genter.